Okay, Eric, ich würde sagen, whenever you feel like it.
hmm Okay. Hi, everyone. My name is Eric Lonergan. We're switching to English now because sadly, neuroscience is a very English-driven field, and I would practice my German and give this talk in German, but the technical terms are simply lacking in German. So this talk will proceed in English. Uh, and hopefully we can have a Q&A in English as well, but I am also able to understand German. If you need to ask a question in German, I will answer in English. So the structure of the talk will be simply a 30-minute lecture, and I will just give it from beginning to end. And then we'll have a Q&A of approximately 15 minutes at the end. And this timing is a little bit loose. If we need to discuss things a little bit more deeply, that's totally fine. I'd like to thank the P.Sc crew for putting this together. This is an amazing initiative, and I hope we can continue to do good work together in the future. Okay, so I am a PhD candidate at the Einstein Center for Computational Neuroscience in Berlin, which is part of the Humboldt-Universität zu Berlin. I am part of the OT Lab, which is the Decision Circuits Laboratory on the Humboldt campus that studies cortical networks that are involved in decision making. And my project is investigating the neural mechanisms underlying psychedelic hallucinations. Hence why I'm giving a talk about psychedelics. So, today we're going to talk about the neurobiological mechanisms responsible for the effects of psychedelics. Now, this is a fascinating and rich and very difficult field, and I'm going to explain why. So it's a very simple question, right? How do psychedelics work? So we have to consider the problem. Consider this plant here, Psychotria viridis, which is known as chacruna. You can extract a compound from this plant that is called DMT, dimethyltryptamine. And chemists understand the molecular structure. It is chemically well characterized. That much is simple. But if you load this into a pipe and smoke it, you end up here. And you don't just end up here. You literally become this. You lose the ability to distinguish between yourself and these maddening kaleidoscopic geometries that are seemingly beyond space and time. So this is clearly quite the problem for neuroscience. How on earth are these perceptual experiences possible? This is a very complicated question to answer. We want to know how on earth this occurs. In order to understand this, we have to take a quick look at the basics in order to then proceed into the mechanisms involved in how the brain is able to produce these kinds of experiences. That's going to be the focus of this talk. In future talks, I can also talk about the potentials and pitfalls of psychedelic drugs as a follow-up. The presentations for those are available. We'll just focus on the basics and the mechanisms today. Okay, so let's dive into the basics first. First, the definition of the word psychedelic. I'm assuming most people are familiar with this. This comes from two words, psyche, meaning mind, and delos, meaning to manifest. So a psychedelic is a drug that manifests in the mind or that makes the mind manifest. Pharmacologically, psychedelics are characterized by the activation of a serotonin receptor, a very specific serotonin receptor known as the 2A serotonin receptor. Serotonin is chemically abbreviated 5-HT, 5-hydroxytryptamine, and the subtype of the receptor is 2A. So when I talk about the 5-HT2A receptor, you'll know that this is the psychedelic receptor. This is the button in the brain that all psychedelics press in order to generate a psychedelic experience. Structurally, psychedelics are very closely related to serotonin, which is why they're able to activate this receptor. Imagine that the receptor is sort of like a lock, and it requires a certain key with a certain shape in order to get unlocked. The natural key for this lock is serotonin. And psychedelics are kind of mimicking this natural key. You can see here in their structures that they're very similar to serotonin. So they're basically able to trick the lock because they're kind of like special copies of this key that are able to fit into the lock in a different way and activate this lock in a different way. There are major classes of psychedelics, 3 of them namely. The first 2 are shown here. We have the phenylethylamines in red, which are chemically related to mescaline, which is derived from the peyote cactus. And then we have the tryptamines down here, which include substances like DMT, which I just spoke about, and psilocybin, which is the other famous tryptamine. There is a third class of psychedelics that is generated when you take the structure of tryptamines and phenethylamines and you sort of put them together and you create a common backbone. It's kind of a mix of these 2 backbones. These are the lysergamides, the most classic example of which is LSD. But LSD is not the only lysergamide. There are many, many, many other lysergamides, potentially infinite other lysergamides as well. An interesting fact is that these different classes of psychedelics have different levels of selectivity for the 5-HT2A receptor. So we call a drug selective for a certain receptor if it binds only to that receptor in the brain and not to any other receptors. So the phenethylamines are relatively selective for the 5-HT2A receptor. They're fairly simple in their pharmacological action. Tryptamines, however, are less selective. They bind to 5-HT2A, 5-HT1A, 5-HT1B, 5-HT2C. Several other serotonin receptors are activated by tryptamines. And the lysergamides are even more complex. They actually activate dopamine receptors, norepinephrine receptors, a variety of different receptors in the brain. So their pharmacology is much more complex. So moving on from the chemistry, let's take a look at why psychedelics are being researched so intensely. We're in the middle of the so-called psychedelic renaissance. And the reason for this renewed interest in psychedelics started arguably with the publication of this study here. Psilocybin produces substantial and sustained decreases in depression and anxiety in patients with life-threatening cancer. Randomized double-blind trial. This was published in 2016. And what they did is they gave psilocybin and psychotherapy to patients who had terminal cancer diagnoses. So these are patients who are on their deathbeds. They're about to die, and they're experiencing extreme depression and anxiety because of their diagnosis. And other antidepressants and anxiolytics just aren't very effective for treating the depression and anxiety associated with the end of life condition. However, when they administered psilocybin to these patients, the effects were remarkable. When they compared the baseline depression with what happened after a single dose of psilocybin, the depression scores tanked. They just dropped very dramatically. They not only dropped dramatically, but with a 6-month follow-up, those patients were still reporting that their depression was gone. So they went into remission sustainably with a single dose of psilocybin. This is completely different from what we observe with, say, SSRIs, which require daily intake of a substance and are relatively ineffective. So this is really revolutionary. And that's not it. They also looked at anxiety, and the results were the same. Psilocybin immediately and sustainably decreased anxiety with a single dose. And when they looked at a metric of the patient's quality of life, they also observed that the single dose of psilocybin remarkably and sustainably increased their overall satisfaction with their life. So this is really, truly remarkable. It's like, how on earth is this possible? How is this behavioral miracle possible? That's an interesting question, but the question gets even more interesting when we consider that psychedelics are able to treat not only depression and anxiety, but they're also able to treat substance abuse disorder, eating disorders, obsessive compulsive disorder, and post-traumatic stress disorder. It keeps going. Psychedelics are actually also useful for treating conditions like asthma or heart disease, known as atherosclerosis. They're also useful for treating arthritis and diabetes. They're also miraculous cures for certain kinds of cluster migraines that are basically incurable with other kinds of medications. So this is extremely impressive and requires some serious explanation. How on earth are psychedelics able to treat so many different pathologies? So we're going to have to deal with this step by step. Well, in the case of asthma, arthritis, diabetes, it's an easy answer because psychedelics are very potent anti-inflammatories. And all of these diseases are inflammatory diseases to a certain extent. With migraines, it's also an easy answer because migraines occur when the blood vessels that feed the brain become too dilated. And so what you have to do is you have to constrict those blood vessels in order to treat those migraines. And psychedelics are very potent vasoconstrictors. So they bring those blood vessels back down into a normal shape and the migraine disappears. So those cases are easy. But what about all these over here? How on earth are psychedelics able to treat so many different psychiatric disorders at the same time, where each one of these psychiatric disorders has typically been seen as an unsolvable problem for psychiatry, each one of them specifically. And now here comes a class of substance that is able to miraculously treat all of these different psychiatric disorders at the same time. So scientists are scrambling to try to understand how this is possible. And we do have some answers, and now I'm going to take you through some of these answers. In order to do so, I would like to establish a framework with the basic biochemistry all the way at the bottom of a pyramid, going through higher and higher levels of analysis in how the brain works until we finally reach behavior and the subjective experience, or the phenomenology, at the top. So first, let's look at molecules and cells at the very bottom of this hierarchy. I've already told you what's going on at the bottom of the hierarchy. The first thing is the activation of the 5-HT2A receptor. This receptor is a complex protein that looks like this monster spaghetti thing, and it's expressed on the surface of many neurons in the brain. And LSD here is able to fit into this binding pocket of the receptor perfectly, and it causes this receptor to change shape in a way that activates it. Once you activate this receptor here, that is schematized in red here, it then triggers a complicated intracellular signaling cascade that leads to a strong activation of the neurons that express this 5-HT2A receptor. And this leads to increased neuroplasticity. And this is a remarkable effect where neurons that are treated with psychedelics suddenly start growing new dendrites, new axons. It's like they come alive suddenly and they start making new connections with the cells around them. You can see this in this study here where they treated neurons in a dish from a variety of different species with either a psychedelic or a placebo here, which is in the color blue. And they simply counted the number of outgrowths from each neuron. in the psychedelic condition versus in the placebo condition. And you can see here that the neurons that were treated with the psychedelics displayed many more new outgrowths in comparison with the placebo condition. So this is an immediate and reliable effect. You treat a neuron with some psychedelics and it immediately starts to grow new dendrites. This neuroplasticity is remarkable and extremely useful. This is known as structural neuroplasticity. This is simply a morphological fact. It's about shapes, right? The neuron simply grows more dendrites. Now, this structural neuroplasticity is just a foundation for what we call functional neuroplasticity. Functional neuroplasticity is the ability for the brain to remodel itself after injury or to be able to adapt to a new situation. You need the structural neuroplasticity. You need neurons to be able to modify their shapes in order to be able to adapt to new situations. So in order to be able to adapt to new functions, for example, memory requires neurons to be adaptable. And that adaptation is simply an adaptation in the structure. So you might be able to see how multiple different diseases could be helped by structural and functional neuroplasticity. If you lose some connections, say with dementia or Alzheimer's or concussion, a hit to the head, that structural neuroplasticity might be able to kick in and allow you the functional neuroplasticity of recovering, say, a speech disorder or recovering movement in a limp arm, for instance. So this kind of neuroplasticity is extremely helpful for all sorts of different diseases. Okay, so we've talked a little bit about molecules and cells and the neuroplasticity that's induced by psychedelics. Now let's take a look at microcircuits. Microcircuits are the smallest units of connectivity between neurons. I'm going to look at one specific microcircuit here, actually more than one. Let's take a look at a brain map of the expression levels of the 5-HT2A receptor. In this map here, the areas that are in red and hot white here express very high levels of the 5-HT2A receptor. There's two main hotspots that are obvious here. The first one is here on the left in the visual cortex. So this might start to give you a hint as to why or how psychedelics are able to induce strong visual hallucinations. If you're simply exciting cells in the visual cortex, it makes sense that there's going to be some visual effects. The other hotspot is in the prefrontal cortex. The prefrontal cortex is one of the most recently developed areas of the brain. This is evolutionarily very recent, and one of the things that distinguishes humans from other primates. We have extremely developed prefrontal cortices. The prefrontal cortices are important for processing social information, for exerting executive control over your actions, long-term planning, also keeping your mood under control. So this becomes relevant for psychiatric disorders. If we actually take this prefrontal cortex area here and we zoom it in, you'll notice here that there is a layer of neurons in the cortex, in the prefrontal cortex, that expresses a high density of these 5-HT2A receptors. In this image here, what's on top here is the skull, and then we have multiple layers of cortex. There's 6 layers in the cortex. Something like 1, 2, 3, 4, 5. Seems like layer 5 is the one that expresses the most 5-HT2A receptors. This layer is very important because it sends connections down to deep brain structures that are involved in all sorts of automatic behaviors. For example, consider the amygdala. The amygdala is responsible for fear and anxiety, and it's a deep brain structure. Normally, the prefrontal cortex keeps the amygdala under control. Let's say you have arachnophobia. You're terrified of spiders. Now, you see a spider, and your amygdala recognizes the shape of the spider and starts to freak out and send panic signals to the rest of your brain. Your heart rate goes up, you start trembling. But then the prefrontal cortex says, no, hang on. That's not a spider. That's just the image of a spider. This isn't actually a dangerous situation. The prefrontal cortex has higher level contextual information about what's going on. And it shuts the amygdala down. It says, no, this is not a situation in which you need to be afraid. That's how this microcircuit operates normally. But in mood disorders, the prefrontal cortex is tired. It's damaged, perhaps by stress, for instance. And what happens is it loses control over the amygdala. So now, even if you see just an image of a spider, maybe you'll have a panic attack. And this is the beginning of, say, a generalized anxiety disorder or panic disorder and other mood disorders as well. The beautiful thing is that the prefrontal cortex expresses these 5-HT2A receptors, and psychedelics activate these prefrontal neurons and normalizes the prefrontal control over the amygdala. So when your prefrontal cortex becomes weak and tired with stress, psychedelics come and they reactivate those neurons. They bring them back to life. Maybe they induce neuroplasticity in the prefrontal cortex, which then allows the prefrontal cortex to regain its strength and to keep the amygdala under control as it should normally, which leads to improved mood. So this is just one example of a microcircuit, right? This is an example of how psychedelics strengthen prefrontal control over the brain's sphere center. There are many other microcircuits that are extremely important for the actions of psychedelics. And of course, we don't have time to go into all of them. I just wanted to mention this specific microcircuit so you could get an idea of how this works. Okay, so we've looked at a microcircuit here. Now I would like to transition to a higher level of analysis, which is the level of large-scale brain networks. Let's talk about what large-scale functional brain networks are. So large-scale functional networks are large ensembles of connectivity and activity in the brain that are responsible for handling different tasks involved in keeping you alive and keeping your perception going. You have many different functional brain networks. For instance, you have the visual network, which simply handles visual information. You have the auditory network that handles sound information. You have the salience network, which is responsible for guiding your attention towards relevant tasks. Anyway, there's so many different functional networks, and usually they're relatively well partitioned. Each one is doing its own task. There's two ways to describe connections in these large-scale functional networks. One of them is a within-module connection. Let's say these are the connections within the visual network. So if you look at these parts of the brain here that are highlighted in blue, so a neuron that's connected in this blue area to another neuron that's in this blue area, that would be an example of a within-module connection. The between-module connections would be, say, crosstalk between different functional networks. Let's say the visual network starts talking to the auditory network. That would be a connection between one of the blue neurons here with one of the purple neurons over here. These different categories of connections will become important in a minute. The functional network that I would like to draw our attention towards is the default mode network. The default mode network involves these areas here: the medial prefrontal cortex, the posterior cingulate cortex, and the inferior parietal lobule. The default mode network has been proposed to be the neurobiological basis of the self. As I've just mentioned, the prefrontal cortex is part of the default mode network, which gives you a hint that they might be influenced by psychedelics somehow, since psychedelics are changing the activity of the prefrontal cortex. The default mode network is called the default mode network because it's kind of like your brain going into idle mode. Like if you stop doing a particular task and you just kind of sit back and relax, and you start to imagine things and you start to remember what happened yesterday and thinking about what you're going to do with your day tomorrow, that's mind wandering, right? So the default mode network is just what your brain defaults to when there's nothing else going on. You just mind wander and you imagine things. But it's also active during rumination, which is when mind wandering gets a little bit out of control. Say in depression, for instance, where you keep ruminating over something that happened in the past and you can't stop thinking about it over and over and over and over again. This involves too much activity in the default mode network. So the default mode network is abnormally activated in conditions like depression. And what's interesting is that the default mode network is disrupted by psychedelic drugs. Because psychedelic drugs bind to receptors in the medial prefrontal cortex and other areas of this network, the default mode network is temporarily destabilized. It sort of loses the ability to do what it's normally doing. And now, if the default mode network is the neurological basis of the self, and it's disrupted, then you'll lose yourself under the influence of a psychedelic. And this explains the phenomenon of ego dissolution on psychedelics, where you lose the ability to distinguish between yourself and everything else that's happening around you. What happens after the psychedelic experience is done is the default mode network comes back together, but in a more regulated way. It's almost like it's being reset. The default mode network goes a little haywire. It is activated too much, and so we shut it down, we reset it, and when it comes back online, it starts to do its job properly without getting abnormally activated. And this helps explain why psychedelics might be helpful for treating depression. All because of those 5-HT2A receptors that are expressed in the medial prefrontal cortex. Another thing that psychedelics do at the level of functional brain networks is they increase crosstalk between the different functional networks. So what happens under the influence of a psychedelic is that the within module connections, these within network connections, they decrease. So the structural integrity of each functional network becomes a little bit weaker. And the between module connections are enhanced. So the different functional networks are now speaking to each other way more than they usually should. It's almost like the networks begin to sort of fuse together, whereas they're usually rather separate from one another. All of a sudden, under the psychedelics, they start to look a lot more similar to one another. This can explain the phenomenon of synesthesia, which is where you're able to hear colors or see sounds. That makes sense. If the visual network is talking to the auditory network way more than it should, there's going to be some cross information between those two networks. And, sounds might start to taste like colors. This could also explain the feeling of everything being connected to everything on psychedelics. If all of the different functional networks of the brain that are responsible for the different elements of your perception are suddenly getting mixed together, it makes sense that you're going to have the feeling that everything is connected to everything else. This also might explain the phenomenon of ego death, where your sense of self is completely lost. Because the default mode network is no longer the default mode network. It's sort of just this massive global connectivity in the brain where each functional network has lost its identity and sort of fused with everything else. If we were to metaphorically or graphically describe this increase in global functional connectivity under psychedelics, we would be able to make a comparison with the placebo condition, where the placebo condition would look like this on the left, with each brain region represented by a little circle, a little node here, and the different connections between them represented by the lines. Now, if we were to add a psychedelic like psilocybin, this increase in global connectivity would look like this massive increase in connections between different parts of the brain. Now, that's a remarkable effect. This should not be taken literally, because this is many steps away from actual measurements performed in neuroscience. This is from a very early paper that was simply trying to conceptualize what it would look like for this increase in functional connectivity to occur. But this is the basic idea, that all of a sudden different parts of the brain are speaking to one another way more than they usually are. And then what would happen after the experience is maybe you would get a more connected brain at the end. Now, to explain this, I'm going to quote David Nichols, who is one of the greatest psychedelic scientists alive, because nobody could describe it better than he can. We speculate that following psychedelic-induced disintegration within local networks, as well as increased global interconnectivity, connections responsible for psychiatric disorder-associated hub failures are disrupted and broken by the emergence of strong, topologically long-range functional connections. Then, as the effect of the drug wears off, networks can reconnect in healthy ways in the absence of the pathological driving forces that originally led to hub failure and disease. In essence, the brain is able to restore its network connectivity to a pre-disease state in much the same way that a computer can be rebooted when its operation becomes sluggish. It's a fantastic quote. Voila, there you go. So we have completed our basic survey of the neuroscience of psychedelic drugs, which now puts us in a position to explain this question mark that would seemingly explain how psychedelics are able to treat so many different psychiatric disorders at the same time. I think a good conceptualization for this is the idea of neural flexibility. Because at the level of molecules and cells, neurons are becoming more plastic. They're stretching out new dendrites, new axons. At the level of microcircuits, we're getting neural flexibility. At the level of long-range functional connections, we're also getting neural flexibility. And this is going to help us adapt to get out of these states where we get stuck in a mood disorder or in a substance abuse problem. Because each one of these disorders could be characterized by a lack of neural flexibility, where you get stuck with some kind of pathological pattern. And the neural flexibility kicks in and allows you to go beyond that. So now I would like to explain some general theories that conceptualize this change. We're going to talk about the so-called free energy landscape of the brain, which is kind of like, it's like an abstract cognitive map of the different states in which your brain can exist. So for example, in depression, you could imagine depression where your brain gets stuck in a valley, right? And these valleys are really deep, very difficult to get out to. Imagine if you put a little ball in here and you try to shake up the system, it's very difficult to escape this valley. And if you would want to, say, traverse this landscape and get from one place to another, you'd have to go over this massive valley over here. So that requires a lot of energy and is very difficult. Now, if we add psychedelics to this, a way to conceptualize the changes to the free energy landscape is that the landscape gets flattened so that the valleys become less intense and the peaks a little bit lower. So if you get stuck in this hole, it's much easier to escape it. This valley is easier to escape, and this peak is also easier to climb over. So now it's much easier to get from one brain state to another. So if you're stuck in a depression here and you flatten this energy landscape, now it's easy to escape it, which helps explain how psychedelics are able to rescue us from states where we get stuck, such as depression, addiction, anxiety, etc., etc. So, putting this all together into unifying theories, I would also like to discuss the so-called thalamocortical gating hypothesis. This is one of the major leading theories for how psychedelics work in the brain at a general level. The idea behind the thalamocortical gating hypothesis is simply that we are opening a perceptual gate with psychedelics. So how does this work? Well, first we have to talk about the thalamus, which is the brain's first relay station that receives sensory information and decides where it goes. Say, the thalamus receives visual information. That's vision. Got to send that to the visual cortex. Here's some sound. I got to send that to the auditory cortex. It's kind of like a switchboard. Decides where the information goes. So you have sensory information that's coming in through the senses and reaches the thalamus. Now, there's a lot of sensory information in the world, too much sensory information. So we have to filter it. And the cortex is responsible for doing that by exerting top-down control over a gate in the thalamus. So it says, thalamus, don't let too much sensory information through because I need to prioritize. I need to focus only on some of the sensory information. So we have a gate that is strict. It's usually closed so that only some information can reach the cortex. Now, if we add psychedelics to this, what happens is the sensory information reaches the thalamus as usual, but the cortex loses its ability to exert top-down control over the thalamus, such that that sensory information now floods the cortex. And if you have too much sensory information flooding the cortex, that might explain some of the sensory alterations that psychedelics cause, like visual or maybe even auditory hallucinations. Basically, psychedelics turn off a perceptual gate that exists inside the thalamus. Now, I would like to emphasize that this is just a theory. This has not been empirically tested yet. This is very difficult to test. But it's one of the leading theories for how psychedelics cause these kinds of perceptual alterations. Another main leading theory is one that we've sort of already discussed. It's called relaxed beliefs under psychedelics, or REBUS for short. It's a very famous theory. The basics of this theory is that our perspective of reality at any given time is a combination of two factors. One is the sensory input. Right? We've discussed that plenty. But another is the prior beliefs that we have about what's going to happen next. You have to combine these two into our real perception of reality. Our perception of reality is not just based on the sensory input. Consider the case where, say, you're trying to find a friend in a large crowd. And you recognize your friend's face, and you know that somewhere in this huge crowd with so many faces that eventually your friend's face is going to pop up and you're going to be like, that's my friend there. And you're looking. Is that my friend? No. Is that my friend? No. Is that my friend? No. Is that my friend? Oh yeah, that's my friend. But then you get a little bit closer and it turns out to be a stranger. But for a second, it really did seem like that was the face of your friend. Why? Well, because you had a prior belief, an expectation that you're going to see your friend's face. And so your perception, your perspective of reality was distorted by your expectation, such that you almost as if hallucinated the face of your friend onto the face of a stranger. So this is an example of how prior beliefs and expectations actually do form the basis of our perspective of reality, or at least part of it. So now that we have this basic framework, now we can explain the theory. The idea, according to this theory, is that psychiatric disorders involve extremely strong expectations, prior beliefs, say about the idea that you're a terrible person. Maybe I think I'm a horrible person and I'm depressed because of that. And the expectation's so strong that I cannot escape it. And it's so strong that the sensory input no longer matters. Maybe there's someone telling me I'm a great person, but I'm not going to believe this person. I'm not going to believe this sensory input because my prior beliefs are so strong. That would look like a free energy landscape like this, as we've discussed, with very deep valleys and extremely high peaks that are very difficult to traverse. A difficult situation. With psychedelics, the situation is theoretically reversed, where prior beliefs are shut down, which allows sensory input to actually influence our perspective of reality again. If we were to plot this on the energy landscape, it would be a flattened energy landscape where it's much easier to move around this cognitive space and much easier to escape your depression into a new state of mind. So there we go. We've discussed each of the slices of this pyramid, and we've also talked about two main theories for how psychedelics work in the brain and how they change perception. That's it. Now I would like to welcome some questions. Okay, you can just go ahead and unmute yourself. You can also drop a comment in the text box, and I hopefully should be able to see that and read it out loud. Let's see if I can find it. Okay, so now I'm looking at the chat. If you want to drop a question in the chat, or you want to say hi with your microphone, you're welcome to as well.
Hi, Eric. First of all, thank you for your talk. It was very interesting. So I was wondering what, and perhaps maybe it's also related to your research, what is the current understanding of neuroscience in why we get these different effects from different psychedelics? For example, with DMT, causing these entity encounters, while that would not be so specific or so common even for LSD.
Yes, that's a good question. And while we don't know the actual specifics for how to answer that question, we do know the beginning of how to answer that question. And the answer is that different psychedelics have different profiles of receptor activation. So, like I explained at the beginning, phenethylamines such as mescaline or 2CB only activate the 5-HT2A receptor and only activate, say, the 5-HT2C receptor. And so they're only really targeting a subset of neurons in the brain. That's not going to lead to these kaleidoscopic journeys through four-dimensional consciousness like DMT. DMT is also a very strong activator of the 5-HT1A receptor, for instance. And one of the ideas behind the 5-HT1A receptor is that it's an inhibitory receptor, and if you activate it a lot, it shuts down the ability of your brain to incorporate new perceptions of the world. And what your brain does is it fills in the blanks. So that is one sort of tentative answer in that direction. Basically, different psychedelics activate different receptors and have different effects. But that's just a pharmacological explanation, right? And the pharmacology is only one piece of the puzzle. Another important question is why, when I take LSD, do I have one experience on one occasion and a completely different experience on the next? That is answered by set and setting. So the diversity, the phenomenological diversity of the psychedelic experiences is to be explained by A, diverse pharmacology, and B, diverse set and setting.
Thank you.
Okay, we have a question in the chat. Could interconnections between brain circuits be simulated or reproduced in another way than taking psychedelics? Yes, but not in humans. So you're talking about causal intervention into the brain in order to try to reproduce these effects. And these kinds of causal interventions are key for science because they're the only real way to effectively demonstrate causality. All we have in human studies is correlations because we can't actually go into a human's brain and mess things up and change things, right? We can't just like cut open your skull and shock certain neurons. We can't do that, right? There are examples of this in animal research, however, where we actually can open up animals' brains and then we can, for example, stimulate neurons directly. Or we can design special drugs that only activate special receptors that have been expressed only in certain neurons. And this has been done before. It's been an increasingly popular technique in psychedelic neuroscience in the last 5 years that's able to recapitulate, to reproduce some of the behavioral effects of psychedelics without a psychedelic at all. So yes, the answer is yes. There is a way to do it, but not with humans. It requires sophisticated techniques in animal research. Okay, here's another question in the chat. I have repeatedly heard researchers say that, for instance, the visual cortex decreases in activity and that this would underline Aldous Huxley's reducing valve hypothesis. Could you elaborate on that and perhaps say a little bit about that research? Sure. Well, first we have to give context about Aldous Huxley's reducing valve hypothesis. This was proposed in the 1954 book, The Doors of Perception. And this is one of the very first written accounts of the psychedelic experience. Now, Aldous Huxley was a very smart guy, and he took a bunch of mescaline and was trying to speculate as to how on earth his experiences were possible. And he postulated that the true state of the mind is the so-called mind at large. That the real natural state of our perception is ego death, total ego dissolution, where we're simultaneously in all points of time and space, right? This cosmic consciousness, that's the default. And that the role of the brain is not sort of to produce consciousness, but to filter this cosmic perception down to something that's manageable for our biological purposes. So this reducing valve hypothesis is actually very similar to the thalamocortical gating hypothesis, right? Both have this idea that there is a gate or a valve, basically, and that normally this valve or this gate is shut, and that under the influence of psychedelics, the valve or the gate is suddenly open. So now that we have a basic idea of what this hypothesis is, now we can actually answer the specifics of your question, which is about activity in the visual cortex. So it turns out that psychedelics do not actually decrease activity in the visual cortex. There have been studies where they put patients, actually I don't think they were patients, I think they were just they were healthy subjects, and they put these patients in an fMRI scanner on ayahuasca, which is a very strong Amazonian brew that has DMT. And what they observed is that when these patients had their eyes closed, that the visual cortex was just lighting up like a Christmas tree. There was just massive amounts of visual activity in the visual cortex without any visual input. Of course, this is a rather coarse metric because an fMRI is not actually looking directly at neural activity. It's looking at what's called the bold signal, which is simply a measure of how oxygenated the blood is going in and out of certain neurons. And it's a proxy for neural activity. So since we can't measure neural activity directly, we're going to measure just the blood oxygenation because neurons that are activated require more oxygen, so more blood gets delivered. So it's possible that there are certain parts or certain microcircuits in the visual cortex that get shut down, and that other microcircuits in this visual circuitry are activated, right? So even though there's research that shows that the visual activity is dramatically increased at the level of fMRI metrics, it's possible that you're still correct. It's possible that certain cells in the visual cortex are actually shutting down. So the short answer to your question is we don't know and we need more research. Okay. So we have another question here. Are the free energy landscapes actually connected to research outcomes? This is a very important question, and the answer is no. There is a problem with this hypothesis. The two authors of this hypothesis are Robin Carhart-Harris and Carl Friston. Robin Carhart-Harris is a psychedelic scientist from the Imperial College London, who is very much committed to the idea that these free energy landscapes are actually connected to clinical outcomes. Carl Friston might disagree, however, because his entire mathematical framework behind the free energy landscape is supposedly not empirically testable, in his own words. So this free energy principle that he proposed, Carl Friston, he described it as simply a framework or a way of thinking about the brain, because there's no real way that we can test the idea that the brain is trying to minimize free energy in this landscape. So this puts a serious amount of pressure on people who would like to claim that the rebus theory is correct, because thus far there is absolutely no empirical evidence that shows that these free energy landscapes even exist, or that they might be changed under psychedelics, or that they might actually correlate with clinical outcomes. Part of my research is actually trying to take a swing at this. So we'll see. Maybe we might be able to show that this is falsifiable. Okay. Another question. How far is the research on psychoplastogens or non-hallucinogenic psychedelics, in the sense that only the neuroplastic effects which theoretically could give rise to these therapeutic effects? Okay. There is a huge debate right now in psychedelic neuroscience between two camps. There's the experienced people, and then there's the neuroplasticity people. The experienced people would like to claim that the psychedelic experience itself is a necessary component for therapeutic outcomes. That you can't get over your depression by simply activating a receptor without the experience. You have to do the work through the experience in order to get out of your depression or your anxiety. And then on the other camp, there's the neuroplasticity people who would like to claim that this is simply a biochemical effect, that you do not need the psychedelic experience in order to get improved therapeutic outcomes. And then there's a spectrum in between. Matthew Johnson from Johns Hopkins University, he proposes what he calls the double whammy hypothesis, which is that it's actually both. There is a biochemical mechanism, say pure neuroplasticity, anti-inflammatory effects, but that the experience is also extremely important for therapeutic outcomes. I think I agree with Matthew Johnson on this, because we do know that there are some purely biochemical influences, like anti-inflammatory effects, that have no experiential, I'll rephrase that you don't have to have a crazy psychedelic experience in order to get anti-inflammatory benefits, right? But that the experience itself from the psychedelic can help you restructure your nervous system's activity in a way that has biochemical influences, right? Because we'd like to think that the cycle between psychology, between our experiences on one side, and between the biochemistry and the matter on the other side is a reciprocal cycle. That what happens on the material level has a psychological influence, but that what happens on the psychological level also has a material influence, right? Think of meditators, expert meditators who are able to get their minds into a state of absolute control. And then you see a massive change in inflammatory biomarkers, for instance. So because of the psychological intervention of meditation, suddenly your body needs less sleep. There's less inflammation. So you think that it's a reciprocal cycle. So I think that this research into non-hallucinogenic psychoplastogens is only going to be partially successful. I don't think that these non-psychedelic psychedelics are going to be as effective as the real thing. But that's just my opinion. We have to see what happens. Okay, last question here. What approaches in research have you learned, and what others are there that you think are promising? Okay, well, that's a very general question. Let me think about that for a second. So the framework that I was discussing previously, which describes our perception of reality as a combination of prior expectations and sensory input, is a very helpful one because it can allow us to computationally model perception in a way that might make these theories testable. So what I do in my laboratory is I work with rats, and thus far nobody has shown that rats actually have a psychedelic experience. And so we did research that proved that these rats have hallucination-like perceptions under the influence of psychedelics. It's a behavioral outcome. Rats are able to hallucinate. Okay, great. But then how is that occurring? So what I do, my job, what I spend all day doing, is in addition to these experiments, I create a virtual rat in a programming language that makes virtual choices in a virtual space, basically. And then I match my model, my virtual rat, to the real rat, right? And then they're sort of analogous to one another. And then I observe the rat on psilocybin. And then I'm going to go back to my model, and then I'm going to tweak my model until my model starts to behave like the hallucinating rat. And if I do this successfully, then I can look at what changed in my model. And I might be able to explicitly uncover the algorithmic changes that are responsible for psychedelic hallucinations. So this is a very useful paradigm because it can be used for computationally modeling perception. So I think that's how I would answer the question of what approaches in research I've recently learned that I think are very promising. Okay. Let's see. Maybe time for one or two more because we're already sort of at 8 p.m. Okay, what answer do you expect? What do you think you are looking for when researching brain function psychedelics? Okay, well now you're putting me against the wall here and asking me like, what do I really think is going on? All right, okay. I've tried to be as neutral as possible throughout this entire discussion. Now you really put me to the wall. So I think that the rebus theory is wrong. I think that what psychedelics are actually doing is the exact opposite, that they're not weakening our prior expectations, that they're actually increasing our prior expectations and decreasing the influence of sensory information on our perception. Because think of the following, when you close your eyes on a psychedelic and you have these complex visual hallucinations, you're not actually getting sensory information from the world, right? Because your eyes are closed. And so what's being projected onto your visual space is not the sensory information that you're getting from the world. It's something that's coming from inside you. Well, what is there inside you other than perceptual information? It's your prior expectations. And they're not prior expectations in the sense that, I think that I'm going to wake up tomorrow and that the sun is going to rise. It's more like the raw mathematical expectations of your visual cortex. It's like your visual cortex expects to see lines and patterns and geometries in the world. And then if we enhance those expectations so intensely, they might be actually projected onto our visual space in a sense that all of a sudden you're seeing the raw fractal mathematical geometry that your visual cortex has evolved to perceive. There is also another implication to this, which is a complicated and tricky question in clinical trials about psychedelics. Currently, researchers are trying to explain, or they're trying to demonstrate in clinical trials, that psychedelics are able to fix depression, that they're able to fix anxiety, that kind of thing. But clinical trials always rely on placebo controls, comparisons to placebo controls, right? And the idea is you need to have a double-blind design in order to guarantee that your study has no bias. The idea is, let's give a placebo to some, and they're not going to know that it's placebo, and we're going to give psilocybin to others, and they're not going to know that it's psilocybin. And then we're going to compare the raw pharmacological effect, but there's a problem. The people who took psilocybin are going to immediately guess, oh yeah, I'm in the psilocybin condition. It's very obvious. And the placebo people are gonna be like, I'm not really noticing anything. I kind of maybe feel a little bit weird, but I don't think I'm on psilocybin right now. What this is called unblinding. And this is really troublesome because it's unavoidable. Like if you take psilocybin, you're gonna know. You're gonna know that you're not in a placebo condition. And this introduces what they call unblinding, or the potential for bias, because all of a sudden you took psilocybin, you know that you took psilocybin, so you expect to get better. And because of the expectation effect in medicine, right, the placebo effect, the outcomes might not be mediated by the pure pharmacological effect of the psilocybin, but simply by your expectation. But this is a very simplistic and old pharmacological model that tries to separate clinical outcomes from expectations. But what if the mechanism of these psychedelics is precisely to enhance your expectations? The placebo effect is an expectation effect. What if psychedelics enhance the expectation effect? What if they make it such that you no longer sort of believe that your depression is going to improve because you took a medication? They make you believe it so strongly that your depression goes away for real. In that case, the pharmacological action itself that statisticians are trying to separate out is actually inseparable from the pharmacological mechanism that they're trying to get their hands on. So it's the same thing. They're trying to separate it, but it's the same thing. And this is a problem that psychiatry keeps running into because of a dualistic Cartesian framework that tries to separate experience from what's happening in the brain. So I think that in order to move forward with psychiatry and with medicine in general, we have to move away from this dualistic framework into a more holistic comprehension of how all of these different levels fit together and actually co-constitute each other. And hopefully psychedelics are going to give the entire field of psychiatry a big trip, such that we can actually move forward not only as human beings, but as a field. Okay, I think we'll stop right there. Thank you very much for your attention and your questions. It was lovely having the opportunity to speak with you. Leonard, would you like to give some final words?
Good. Thank you so much for this amazing presentation, for the surprisingly in-depth account of the underlying mechanisms of psychedelic action, and also this just very great neuroscientific introduction. I really enjoyed it. I think we covered so much, much more than I had expected. So I'm really stoked about that. And also, I'm really happy about hearing about your own research. This came up in the questions, and it was my personal question as well. I wanted to post, but then it was answered already. That's super fascinating. Thank you so much. And I think we'll upload this talk to YouTube for everyone to check out remotely as well. And then we'll see each other for the next lecture in 1 month at its latest. Yeah, I think that's it for today. Thank you all for being here, especially to you, Eric, and see you around next time.
All right, everyone. Have a great rest of your day. Bye-bye.
Take care.